Today, the speaker is going to be Evani Radiya-Dixit. So first of all, let me introduce Evani. So she is a senior at the Harker School, and her interests in science were sparked from doing projects at home at a young age, including building an algal photobioreactor in her own backyard. Since 7th grade, she has worked on independent research projects in various STEM fields, ranging from biochemistry to aerodynamics. Over the past 3 years, she has focused on using computer science in cancer research, employing tools such as natural language processing, machine learning, and statistical methods to improve diagnostics and treatment. Evani has had the opportunity to compete in various research competitions and has placed in the Siemens National Competition. Her recent project was conducted at the Harvard Medical School under Dr. Andrew Beck. She studied genomics data for cancers of the lung, pancreas, and rectum, and was recognized as a Regeneron Science Talent Search finalist this year. Outside research, Evani is the co-president of the Harker Future Problem Solving Program. She received an NWIT grant to teach girls programming using MIT App Inventor. In her spare time, Evani enjoys running, writing, and singing. In college, Evani plans to pursue computer science and entrepreneurship. Using technology, she aspires to work on global challenges in healthcare and education. Today, Evani will be presenting her project, which is titled Identification of Diagnostic Biomarkers and Therapeutic Cancer Targets Across Adenocarcinomas Using Genome-wide DNA Methylation Analyses. Now, without further ado, it is my pleasure to welcome Evani Radiya-Dixit to the stage. Let's give her a welcome applause.
Thank you for the introduction. So my name is Evani, and my project work involved the identification of diagnostic biomarkers and cancer therapeutic targets across adenocarcinomas using genome-wide DNA methylation analyses. So every year there are about 596,000 cancer deaths in the US, and pathologists misdiagnose from 10 to 20% of cases. And the rate of risk of recurrence, even if they are able to treat a tumor, ranges from 30 to 55%. So these kind of served as my motivation for my research work, with the objective of identifying sites on the genome that can be used for diagnosis, as well as identifying proteins and pathways involved in tumor progression, and that way these can be used for drug development. So I studied the 3 deadliest adenocarcinomas, and adenocarcinomas are cancers that form in mucus-secreting glands. So I studied the cancers of the lung, pancreas, and the rectum, and these account for about 42% of all cancer deaths. And this was in the US in 2016, and you can see that over here. So what I did in my research work is I used DNA methylation as a window into cancer. What happens during methylation is that a methyl group, CH3, attaches to the genome at a CpG site, and this is a cytosine followed by a guanine. And when the methyl group attaches, that can alter the gene expression. That can either lead to increased gene expression or reduced gene expression. And I studied the methylation levels and compared them between normal samples and tumor samples. And you can see the difference in methylation levels in this figure on the right, where the open circles represent unmethylated cytosines and the closed circles represent methylated cytosines. And these are differences in methylation patterns across the genome. So my approach is an advancement of existing work. For one, I studied more regions of the genome. So traditionally, only the promoter region has been studied, and you can see that over here. But I studied these other regions of the genome, including the intron region and the extron region. Second, I accounted for biases using my pre-processing methods, and I'll talk further about that later. And third, I studied the 3 deadliest adenocarcinomas, and there's limited knowledge about the commonalities among these cancers. And it's really important that we identify a common drug that can be combined with personalized treatment due to the high risk of recurrence. So to perform these analyses, I used methylation datasets from the Cancer Genome Atlas, TCGA. And these methylation datasets given as beta values between 0 and 1, where 0 is completely unmethylated and 1 is completely methylated. And the methylation levels were given at about 490,000 sites. And here at the table, you can see the number of samples that were given for the different cancers: lung, LUAD, pancreatic, PAD, and rectal cancer, READ. So the data was collected using 2 different probe types, where type 1 probes are 2 probes with 1 color, and each of the probes measures one probe measures the unmethylated intensity, and the other probe measures the methylated intensity. And type 2 probes have 1 probe with 2 different colors, where the 2 different colors distinguish between these 2 intensities. So I developed a comprehensive pipeline to process the methylation data, and it involved 3 key steps, the first step being pre-processing, and this was to ensure data quality and to avoid misleading results. So for pre-processing, I first filtered the data, removed sites at which the methylation detection p-values were very high, as well as those that were in repeated DNA sequences. Second, I performed intrasample normalization. And as mentioned earlier, the data was collected using 2 different probe types. So I had to adjust the methylation distribution of the second probe type to that of the first probe type. And then finally, I performed batch effect removal. So the data was collected using different batches. And what I found is that within each batch, the methylation levels were similar, and so I had to account for this confounding variable. And you can see here at the figure on the left, where each line is a different sample and the colors are different batches. And what you see is that, for example, the yellow lines are clustered together, which indicates that they have similar methylation levels. But after batch effect removal, we no longer see that bias. After pre-processing the data sets, I then performed differential methylation analysis. So here's where I found the sites that distinguish between 2 different groups. I first found the differentially methylated loci, DMLs, that distinguish between normal samples and tumor samples. So here are heat maps showing the DMLs for each of the cancers, for lung, pancreatic, and then rectal. And you can see that the rows are the different samples and the columns are the different probes. And the methylation levels are shown as a gradient between blue, which is a beta value of 0, and 1, and red, which is a beta value of 1. And you can see that there's a very clear distinction between the normal samples and tumor samples, indicating that these are the sites where the methylation levels are really very different. And then after performing this for each of the cancers independently, I then found the common sites across the 3 adenocarcinomas. So I found 604 DMLs across the 3 adenocarcinomas, and actually 64% of these sites were not within the promoter region. So this is really important because it shows we need to be looking at these other regions of the genome because they're contributing to, or could be contributing to, tumor progression. I then performed similar analyses to identify the differentially methylated loci that distinguish between tumor clusters. So I first clustered the tumor samples into 2 subgroups, and you can see that here with the sidebar of green and orange. And the methylation levels between these 2 subgroups differs a lot, and this is really important to show the tumor heterogeneity. So even though we're able to distinguish between normal samples and tumor samples, there's still much variability within the tumor samples. So these are just the number of DMLs that I found for the 2 sets, as well as the sizes of the 2 clusters. So having found these 2 sets of DMLs, I next wanted to find the biological implications of this information. And what I did is I performed gene enrichment analysis. And this is where I was able to find the proteins and pathways involved in tumor progression and tumor heterogeneity. And I found the corresponding sets for the 2 sets of DMLs that I identified. The way that gene enrichment works is that using the sites, I was able to map them to genes, and then using current research about existing pathways for particular protein functions, I was able to find the pathways that are enriched. And I did this for the 3 adenocarcinomas independently, And I also accounted for the fact that there is a greater proportional differential methylation with a greater number of sites per gene. So with more sites per gene, you can see that there is more methylation, and I accounted for that bias. And then I found the common sets for the 3 adenocarcinomas. So I found 33 common sets for sites that distinguish between normal and tumor samples. And then I found 12 common sets that distinguish between the tumor clusters. And you can see here visually that I was able to find 4 proteins and pathways involved in these common sets. And they are very similar between the sets that distinguish between normal and tumor, as well as those that distinguish between the tumor clusters. And the distribution of these 4 different proteins and pathways is also pretty similar. And the way that these can be used is that we can develop new drugs to target these proteins and pathways. So the first protein I found was the polycom group proteins, and what we can do is we can target the subunits of these proteins with new drugs. Second, I found the extracellular matrix proteins, and these are outside the cell, and they contribute to the structure and provide support for surrounding cells. Third, I found G protein coupled receptor proteins, GPCR proteins, and these are also known to be involved in cell proliferation. And so a disrupted protein can thus lead to uncontrolled growth. And then I found epidermal growth factor signaling that this pathway was affected, and the certain specific pathways here are involved in cell proliferation. So what is the significance and what are the implications of my work? Well, for one, there are a lot of benefits that can be used for diagnosis. So I found 604 sites that distinguish between normal samples and tumor samples across the adenocarcinomas. And out of these, 64% are outside the promoter region. And these are for the 3 deadliest adenocarcinomas. So what we can do is we can induce methylation at these sites to confirm them as diagnostic biomarkers. And we can do that using a repurposed CRISPR-Cas9 system. And with that experimentation with living animals, we can then use these sites for blood-based diagnosis, as well as better understand the changes in the protein levels, and then use protein expression in serum-based diagnosis. There are also several implications for treatment. So I identified the 4 proteins and pathways, and we can develop new drugs to target those. So previously, this study had identified or developed a drug that can target one of the proteins that I identified. And so similarly, drugs can be developed to target the 4 proteins and pathways. And this is really important that it's a common drug because of the high risk of recurrence. And when we combine that with personalized treatment, then that can really reduce the risk of cancer. Other implications besides diagnosis and treatment include clinical trial designs. So currently, many clinical trials are designed and patients are stratified based on cancer subtypes. But as we saw in the heat map within the tumor samples, there's a lot of heterogeneity, and we're able to classify those different groups based on the methylation levels. So I propose using methylation-based subtypes as a much better approach to identifying potential treatment for in these clinical trials. And then finally, there are also applications in computational research. So my pipeline can be used for data sets outside of TCGA, as well as for other adenocarcinomas. And we can also use it to identify common biomarkers and therapies for sarcomas, melanomas, and lymphomas. Finally, I'd like to acknowledge my mentors, Dr. Beck and Mr. Glass of Harvard Medical School, as well as Dr. Tim Trish of the USC Cancer Center. And I'd like to thank Mr. Spenner and Ms. Chetty of Harker School for all their support. Thank you.
Hi everyone. So thank you. Let's give Evani another round of applause, please. Okay, so we're running a little bit late, so we're only going to answer 2 questions today. So the first question is, how do you get access to all of these resources?
So in terms of the data, all the data is available online. So I used it through the GDAC portal, which has access to the Cancer Genome Atlas data. And there's a lot of genomics data that's available, as well as, I think, biopsy images that are available online if you're interested in bioinformatics work, as well as like learning resources. I think a lot of information is available online, and that's really where I learned how to process the data set and looking at publications, how they process the data set to better understand how to identify genomic sites and the potential therapies.
Okay, so we're gonna use this mic. So the other question is, how did you get an idea to do research in this area, and how was it supported by Harker?
So in terms of how I got into this field, so I've actually been doing cancer research for 3 years, but each year I've kind of delved into a different area and used different types of data. And that really allowed me to develop the skills to learn how to process data quickly and explore publications in terms of science research in general. But I particularly was interested in epigenetics because it involves environmental changes to the genome as compared to genetic mutations. And that aspect really intrigued me, and I was interested to learn how that can contribute to tumor formation. And then the second part of the question was? How did Harker help? So I think there's a lot of support with the research program. So I actually took the research class with Mr. Spenner, and I found that very useful to allot a time every single day or every other day to working on my research. And then also I think there was generous support in terms of like reading the paper. So I got feedback from Mr. Spenner, as well as had a conversation with Ms. Chetty about it. And that was really useful to better understand my discussion and conclusions.
Thank you, Evani, for a great presentation.